This is Ella, a biochemist developing a new vaccine candidate. Her experiments include characterizing thousands of antibody candidates against a target protein and determining their binding kinetics. She's using surface plasmon resonance, or SPR, to analyze these interactions in real time without the pitfalls of labeling her samples. SPR is an indispensable technology for Ella's work in drug discovery. In SPR, a beam of light bounces off the back surface of a sensor chip at a particular angle and into a detector. When probe molecules linked to the biosensor surface bind with free molecules in solution, it causes an angle shift of the reflectivity curve. By measuring the time-resolved shifting angle, scientists can track granular kinetic behavior, including rates in association and dissociation between molecules. But with her lab's current SPR setup, Ella's limited to running just a few samples at a time. As always, she has a tremendous amount of work to complete on a tight deadline. Her SPR system isn't flexible enough to handle her current workload. And if she pushes for more throughput, she's going to lose sensitivity. That could be disastrous in these early stages of drug development. Resigned to missing more family and social events, Ella is preparing herself to work several nights and weekends, even though she knows she'll never be able to complete all the samples in time. It doesn't have to be this way, though. The Bruker SPR64 is an all-new, flexible system offering 64 sensor spots in an 8x8 array. The fluidic setup allows the handling of up to 8 different buffers in parallel, leading to high-end multiplexing on a single sensor chip. With a rotating manifold that changes its injection direction, the SPR64 allows the full kinetic characterizations of 8 targets per injection, while reducing the final target consumption to 1/8 compared to common SPR systems. All this means Ella has fewer injections, shortening her runtime and giving her results faster. And since the SPR64 is capable of measuring crude samples from cell supernatants or bacterial lysates, Ella spends less time pre-processing or purifying samples. The SPR64's simple and robust microfluidics doesn't compromise on sensitivity or high throughput. Plus, with low sample consumption, Ella doesn't have to worry about wasting reagents. With a full range of applications, it can even use external automation or be easily integrated into a fully automated environment. The difference between other smaller machines and the Bruker SPR64 is more than just scale. It's like having a single orange tree versus an orchard. With one tree, you can't expect more than a modest crop of fruit to be enjoyed with friends and family. When production grows to an orchard, though, the abundance can cover commercial scale distribution, processing options, and higher income. It means more results, more reliability, and at a higher rate. You can finally have flexibility, throughput, and sensitivity. It's not just Ella that's getting more work done with the SPR64. It's flexible enough to handle applications in kinetics, classification and specificity, fragment screening, multiplexing and condition screening, epitope characterization, quantification, thermodynamics, mode of action studies, and more. Already well known for our wide catalog of laboratory instruments and patented workflows, we're proud to be extending our offerings with the most complete surface plasmon resonance system on the market. You have science to do, and Bruker has the tools to help you do it faster. Visit Bruker's website to see how the SPR64 is designed to succeed and accelerate your drug discovery.